So first, I wanted to say thank you for inviting me to give this talk. And today I'll be talking about false positive IRESes, mRNA annotation errors, and a paradigm unshift in mammalian development. And I'm going to be talking specifically about a project that we worked on for the past 2 years during COVID. And I'd like to highlight Gemma May right here. She performed all of the experiments I'll be talking about today. So gene expression is controlled by a wide range of mechanisms. We have transcriptional control, alternative splicing, nuclear export, and translation. While all of these levels of control are very important for gene expression, our lab focuses on translational control. And translation as a regulatory mechanism often offers a rapid response to stresses or cellular development by directly regulating protein expression and without impacting these other levels of control, for example, splicing or transcription. Thus, translation is important in defining the proteome, controlling cell growth, and maintaining cellular fitness under stress. Eukaryotic translation is broken up into 3 parts: initiation, elongation, and termination. However, translation is primarily limited by initiation, where the pre-initiation complex or ribosome identifies a start codon and commits to protein synthesis. Typically, mRNA translation follows a cap-dependent directional scanning model. First, the EIF cap binding complex binds to the 5' cap of the mRNA. The 40S pre-initiation complex, which is made up of several initiation factors, is then recruited to the 5' cap. It then scans the transcript leader in search of a start codon. In some cases, cap-dependent translation is repressed, and you can get cap-independent methods for translation. For example, viruses use internal ribosome entry sites, or IRESes, which are structures that can directly recruit the pre-initiation complex with the help of different factors. So this was first identified in viruses as a way to hijack endogenous mRNA translation, but maintain viral or translation of viral transcripts. In the last 2 decades, there have also been studies done of cellular mRNA IRESes. In 2015, Zhu et al. Tested developmental Hox genes in mice, known as Hox genes. So they tested 5' transcript leaders for potential IRES activity. And to do this, they cloned different transcript leaders in a PRF plasmid with a CV40 promoter between this Ranilla luciferase and Firefly luciferase, so R-Luc and F-Luc. So they cloned the transcript leader between these 2 bioluminescent proteins and then transfected the plasmids into a mesenchymal cell line. And the idea of this is the F-Luc should be translated using cap-independent methods, while the R-Luc should be translated using the cap-dependent methods. So from this, they measured the different F-Luc activities for all of these Hox transcript leaders, and they found several of the Hox transcript leaders seem to have significant F-Luc activity. So for example, the Hox9 transcript leader had the highest F-Luc activity in this bicistronic reporter assay. They then deleted different parts of the Hoxa9 transcript leader to identify which region of the transcript leader is truly important for the F-Luc activity. So they deleted different parts of it and found that the 3' end of this transcript leader was most important for F-Luc activity. So they concluded that the 3' end is a key part for putative IRES activity. Later in the paper, Barna and Das also generated in vitro Hoxa9 RNA and probed its structure using shape probing in vitro. They identified these 4 different domains, which they labeled P1, P2, P3, and P4. So this was the 3' end of that HOXA9 transcript leader, which was shown to impact F-loop activity. They then deleted different parts of this structure to identify the key component of the putative HOXA9 iris. They did this using bicistronic reporter activities. So in this corner, we see the results from deleting different regions of this structure. So they found that the P3 and P4 domains, or those stem loop structures, have the greatest impact on F-loop activity. Building upon earlier work, Lepik et al. Further analyzed the structure of the proposed HOXA9 iris element. To do this, they mixed HOXA9 RNA with the small ribosomal subunit in vitro at 4 degrees. So from this cryo-EM structure, they found that the P4 loop of the HOXA9 iris seems to be interacting with the expansion segment 9S from the ribosome. So from this, they thought that the P4 loop may be very important in having significant F-loop activity from those bicistronic reporter assays. So to identify which region of this P4 loop was very important for this interaction, they again used bicistronic reporter assays to delete different regions of the P4 stem loop. And here, anything with a plus means that there was significant F-loop activity, and if there was a minus, that means they saw a significant reduction in F-loop activity. So first, they mutated the 5' half of this stem loop, as well as this loop structure, and they found that it did not impact F-loop expression. They also mutated the 3' end and saw that that did in fact impact or reduce F-loop activity in their bicistronic reporter assays. However, compensatory mutations, which rescued the structure from that 3' stem, did not save or rescue F-loop expression. So from these studies, they concluded that this P4 loop plays an important role in the putative iris activity for the HOXA9 transcript leader. Lepik et al. Also developed a pull-down assay to identify other putative mRNA and ribosomal expansion segment interactions. To do this, they engineered, or they took yeast ribosome and engineered a humanized version of the ribosome. So basically, they used the human expansion segment 9S to test which mRNAs would be linked or have propensity to bind to this expansion segment. So they also introduced a flag, which will later be used for the pull-down assays. So they mixed these ribosomes with fragmented mouse embryo mRNAs, and then ran a pull-down assay to selectively purify the interactions of these mRNAs with the humanized ribosome. And after that, they can run RNA-seq to identify which mRNAs bound to the expansion segment. And from that, they found that a ton of G-rich mRNAs had propensity to bind to that expansion segment ES9S. So a recent study used the conservation pattern of the HOXA9 transcript leader to identify other ultra-conserved transcript leaders. And to do this, they used FAST-CON score, which is a conservation score across nucleotides. So using the HOXA9 transcript leader pattern, they found 589 genes in the mouse genome that seem to match the HOXA9 transcript leader conservation pattern. And from this, they used Go enrichment and found that these genes were enriched for mammalian development. The Barna group then tested over 200 of these hyperconserved elements using a bicistronic reporter assay in 6 different mouse cell types. So in the graph to the left are the transcript leaders ranked based on their bicistronic reporter activity. So that's shown on the y-axis, and just the rank is on the x-axis. And just for reference, the 2 viral irises which were used as control are shown in this green box, as well as HOXA7 and the HOXA9 putative iris. They also plotted the reporter activities using this violin plot. So here on the x-axis is also the bicistronic reporter activity. And when looking at this violin plot, they saw these two distributions, which seemed to be of separate populations. And they separated these distributions into what they called active and inactive. So the active transcript leaders here, they concluded that they could have potential IRES activity. Meanwhile, the inactive ones are less likely to have FLuc activity. So in this active group, they found 90 different transcript leaders. Combined, these studies propose a new paradigm for mammalian translational control because they suggest that IRESes are common in mammalian genes. They further identified that developmental genes are regulated by IRESes. These studies would also explain another function of ribosome expansion segments. However, there are still some questions that remain. For example, why would Hox genes use the less efficient IRES method for translation? And what is the reason for the HOXA9 transcript leader having so many UORFs within the upstream region? Additionally, there was previous work that actually showed that the human expansion segment 9S did not actually support IRES activity. So over COVID, we were reading the cryo-EM paper, and we noticed that this HOXA9 P4 loop interacts with this expansion segment in a kind of a kissing loop formation. So looking more into this structure, we saw that the HOXA9 P4 region, so this is highlighted in blue, has a G-rich segment. And that would match what was identified via Velcro IP, where G-rich mRNAs had propensity to bind to the ES9S expansion segment. But when looking back at this structure, we saw that their bicistronic reporter data didn't quite fit this proposed structure because they could delete the loop of this HOXA9 P4 structure and also the 5' end, and that wouldn't impact FLuc activity. But deleting this 3' stem loop was the only part that actually decreased the FLuc activity in the bicistronic reporter assays. So this didn't quite fit the model they proposed, so we decided to examine the HOXA9 transcript leader structure a little more. So we looked back at their original paper and identified the structure, which identified the structure of the HOXA9 transcript leader. So they originally used shape probing and found that this P3 and P4 region was important for bicistronic reporter activity. They also concluded that the IRES must be conserved because the zebrafish HOXA9 transcript leader, shown here, also seemed to drive FLuc expression in bicistronic reporter assays. So from this information, we first examined the structural conservation of the putative IRES region. Here, I used the RNA structure prediction program to test the conservation of this IRES region or putative IRES region in mouse, human, and zebrafish. So first, using RNA structure prediction program, I modeled the structure of the putative mouse IRES. So that's shown right here. So each black line indicates a base pairing between nucleotides. And I ran this program 10,000 times, and the probabilities are shown under these red lines. So the probability of the P4 loop occurring is 96% out of those 10,000 runs. So I then ran the predicted structure for the human region, and we do not see the same formation of the P3 structure and don't see such high percentage for the P4 loop either. And now looking at the zebrafish on the right, we don't see the P3 or P4 loop happening at all. So this data suggests that the proposed IRES was not actually structurally conserved. To further explore the evolutionary conservation of the putative IRES structure, we compiled homologous regions of the HOXA9 transcript leader for 230 vertebrates. So for this, we had an alignment of the putative HOX IRES region, and using RScape, which is a structure algorithm, it looks for covariation. So the reason to do this is if we see significant covariation amongst the alignment, we could conclude that the structure is actually conserved. So for example, if you had a mutation which changed a nucleotide, you would see a compensatory mutation which rescued the structure. So using the program known as RScape to test for structure covariation, we found these results to the right. So I'll break it down for you. First, the y-axis is showing the significant base pairs, and the x-axis shows the expected pairs, so the e-value. And the blue line here shows the results of the prediction program algorithm at different e-values. So for example, at the significant e-value of 0.05, we had an alignment that had the power to detect 10 significant base pairs. However, we only saw 0, so we saw no significant base pairs for evidence of covariation. And only when I raised the e-value to 20, so that's when you have an e-value of 20, you expect 20 base pairs by chance. We only get 10 significant base pairs. So this modeling shows that there is no significant covariation amongst the 230 vertebrate alignment. So from our analyses, we saw no structural conservation of the putative HOXA9 IRES. So next we looked at IGV with some public data sets. So we found that the HOXA9 transcript leader was shockingly misannotated. So this here is the HOXA9 gene, and here we see HOXA10. So just to orient you, this would be the 5' transcript leader that was annotated for HOXA9, the putative IRES region, which is highlighted in pink, and then the 3' region. So using ENCODE RNA-seq data, we see that the RNA-seq doesn't cover this transcript leader at all. Instead, it covers the gene very close to the coding region. There were also transcription start site data from Ivanov et al. Published in 2022, which found or mapped the transcription start sites much closer to the HOXA9 coding region. So there was no transcription start site at this annotated 5' end. There was also ENCODE data and EPD data for promoter elements, and the promoter elements aligned with this mapped transcription start site and would support a shorter version of the HOXA9 transcript leader. There's also RNA-Pol2 data, which again aligned with this promoter element and transcription start site. Looking at full cDNA reads, we see that there's much shorter, the transcript leader mapped is much shorter than the one annotated here and used by the Barna lab for their original HOXA9 IRES identification. And looking further, there are some fusion reads actually that merge the HOXA9 with upstream genes, for example, this HOXA10, HOXA9 fusion. So this supports a shorter version of the HOXA9 transcript leader and shows that this longer transcript leader is actually misannotated. So the putative HOXA9 IRES that they were testing is actually an intron and not a 5' transcript leader. And this would explain why there was no conserved structure when we were looking at the RNA structure program or RScape. So consistent with this, Ivanov Ivanov et al. Found that the HOXA9 transcript leader is also misannotated using qPCR. So Ivanov et al. Used qPCR in mouse somite RNA to see if the proposed IRES sequence was actually amplified. To do this, they had primers at the putative IRES, and they also had primers of the coding region. And as a control, they also ran qPCR on a synthetic HOXA9 RNA, which contained the putative IRES sequence. So this is their data or their results from the qPCR, and they found that the IRES region was 200-fold less abundant than the coding region. And then as a control, they ran the qPCR on the synthetic HOXA9 mRNA and saw that the IRES could in fact be amplified. So that was not the issue when they were looking at this total somite RNA. So this also shows that the putative HOXA9 IRES is not transcribed. In 2017, Barna's group performed ribosome profiling and RNA-seq from mouse embryos. And so this is their data from neural tube, these top two graphs. So here we don't see any ribosome or RNA-seq pileup in this putative transcript leader or the transcript leader tested previously. Instead, we see support for a much shorter transcript leader. So this data shows that the actual HOXA9 transcript leader is much shorter than the ones previously tested. And this profile kind of matches the HOXA10 gene, where you have some data over the 5' UTR, have a dip during the intron, and then some more data during the coding region. And just to zoom in on this data a little bit closer, so here again is the ribo-seq data and the RNA-seq data, and they align with the corrected HOXA9 transcript leader isoform. And this also actually supports the UORF identified in Ivanov et al. In 2022. And the more we worked on this problem, the more we realized that we weren't the first ones to notice the issue of the HOXA9 transcript leader. A lot of these issues were brought up in PubPeer in 2014. And I can share this link later on after the talk if you're interested in reading. But there was also a review in 2017 that stated that the HOX transcript leaders are probably much shorter than what is shown in public data. So far, we found that the putative HOXA9 IRES is not structurally conserved. And we found that the HOXA9 transcript leader is misannotated. But what then explains the reporter results from Zhu et al., which saw F-loop activity of these HOX transcript leaders? So bicistronic reporters are commonly used to test IRES activity. A bicistronic reporter assay consists of typically Ranilla luciferase and Firefly luciferase, so R-Luc or F-Luc, and then the potential transcript leader that has IRES activity. And by cloning that between the R-Luc and F-Luc, you can later compare the R-Luc to F-Luc activity for a bicistronic reporter score or a potential IRES score. And the idea is that this F-Luc is translated via cap-independent methods. And just for another schematic, this is an example of the two different proteins being made. However, the reliability of these bicistronic reporter assays is subject to debate because they often result in false positives. For example, if the transcript leader tested, so that's the transcript leader between the clone between the Ranilla and Firefly luciferase, if that contains a promoter element, you can actually generate this monocistronic F-Luc transcript. So you would get F-Luc expression even though it was just using the cap dependent model. So it was not using the IRES cap independent system. Instead, it's just using canonical translation. And one way to check for this is to delete the SV40 promoter. And if you still get F-Luc activity, you know that you're generating monocistronic F-Luc. Second, there could be splice sites or cryptic 3' splice sites within that transcript leader that you're testing. So if the 3' splice site is present in a transcript leader, you can also splice out this R-loop and again generate a monocistronic F-loop transcript. And one way to check for this is to do RT-PCR to find which transcripts you're actually generating. And you can do this if you know the 5' splice site. But 5' ends are not always easy to identify. Previously, Lemp et al. In 2012, they found that the PRF plasmid actually generates cryptic upstream promoters and can create these aberrant transcripts. So they found that the PMB1 ORI can generate these random spliced artifacts. So these are other ways where the 5' end may not be what you would expect typically from the bicistronic reporter assays. And added to this complexity is the mammalian transcriptome, which inherently contains a lot of introns, promoters, and splice sites. Now, thinking about these controls and how the HOXA9 transcript leader was misannotated, we hypothesized that the annotated HOXA9 transcript leader is actually a promoter. And to investigate this, we ordered the same bicistronic reporter that Barna used, but instead we deleted the SV40 promoter. And between R-Luc and F-Luc, we cloned in the HOXA9 full transcript leader, also the HOXA9 full transcript leader, but reversed. We also had a version where only the HOXA9 putative IRES region was included, or the putative HOXA9 IRES region with it reversed. We then transfected these plasmids into mouse embryonic cells. Then measuring the F-Luc to R-Luc activity, we saw that there was a significant F-Luc activity in the transcript leader and in the IRES, even though the SV40 promoter was deleted. So this is showing that there was independent promoter activity in the transcript leader we tested. And when you first look at this, it might seem that the IRES isn't showing a significant amount of F-Luc, but when we looked at the raw data, we actually saw that the IRES transcript leader region actually generated spurious R-Luc, and this might be due to the PMB1 ORI. So when comparing those ratios, that would mean the denominator has changed. So that's why the F-Luc is actually much greater than what is seen in this ratio. We also ran these experiments with the human HOXA9 transcript leader and saw the same conclusion, where the transcript leader and IRES region had independent promoter activity. And we're not the only ones to show this. Earlier this year, the Dever and Green labs showed that the HOXA9 transcript leader induced expression in mammalian tissue culture. So this is again the bicistronic reporter assays. As a follow-up to our reporter experiments, we performed 5' race on the cells after transfecting the plasmid. So we mapped the F-Luc transcription start sites to the annotated promoter sequence. So this supports a much shorter version of this HOXA9 transcript leader. So the previous HOXA9 transcript leader tested was actually misannotated, and the corrected transcript leader aligns with annotated promoter data and our transcription start site mapping. Finally, we tested other HOX transcript leaders using the bicistronic reporter assay, and we found that all transcript leaders which were previously identified to have significant F-Luc expression exhibited independent promoter activity. Meanwhile, the transcript leaders without putative IRES activity did not show the same independent promoter activity. And the raw data for the graph above is shown here below. And consistent with our reporter results, we saw that the putative irises, the other putative irises were also incorrectly annotated. For example, here is an IGV screenshot of the HOXA9 transcript leader. So in Zhu et al., they annotated this much longer HOXA4 transcript leader. Meanwhile, the RNA-seq data, promoter elements, and transcription start site data all map to this much shorter annotated HOXA4 transcript leader. Here's an example of the HOXA7 transcript leader, which is misannotated, but has RNA-seq data and RefTSS data that support a much shorter version of the transcript leader. And HOXA11, which was also annotated in the Zhu et al. Paper, is also misannotated. Instead, there's RNA-seq coverage and transcription start site data, which would align with the annotated shorter HOXA11 transcript leader. So far, we found that the HOXA9 structure is not conserved and that the transcript leader contains a promoter and is misannotated. But what would explain the reporter or the F-loop activity they saw when they deleted the 3' end of this P4 stem loop? So why does deletion of this region impact the bicistronic reporter activity? So we next looked more closely into this 3' end of the HOXA9 P4 region. And using FIMO, we actually found that this 3' region contains an E-box motif, CACGUG, which is recognized by USF1 and USF2 transcription factors. And this motif is also recognized by 30 other transcription factors, including MYC. So we next generated a sequence alignment of vertebrates for the P4 stem loop region of this HOXA9 transcript leader. And we found that the E-box region, the CACGTG motif, is almost universally conserved, as well as the CAAT box core promoter element. Meanwhile, the G-rich region, or this loop region, which has propensity to bind to the expansion segment, is not similarly conserved. Interestingly, previous work showed that the USF2 transcription factor binds upstream HOXA9 in the human transcript leader. And another study found that co-depletion of USF1 and USF2 actually depletes or decreases expression of HOXA9 expression. So next we looked at public ChIP-seq data to see if other transcription factors bind the HOXA9 region. So we found that USF2, USF1, MYC, and other transcription factors bind exactly where this E-box motif is located in this HOXA9 region. And here the colors just represent different significance of the ChIP-seq peaks. So next we tested the hypothesis that the E-box motif contributes to promoter activity of the HOX genes. And to do this, we constructed two different plasmids. One plasmid had the R loop as a control, and the other plasmid had the F loop with the putative iris insert, or now we know the promoter element. So we had the HOXA9 transcript leader and the HOXA9 transcript leader with the E-box mutated. We also tested the HOXA9 putative iris region and the HOXA9 iris putative iris region with the E-box mutated. And then we compared the F loop to R loop to get bicistronic reporter activity. So that's what's shown here on this graph, the y-axis. And on the x-axis is the different levels of F loop for the different plasmids. So we had the HOXA9 transcript leader, the iris region, and also for the human isoform or the human version of the HOXA9 transcript leader, and also the putative iris region. And here we saw wherever the E-box was mutated, we had a decrease in F loop expression. We also used co-depletion of USF1 and USF2 to see the impact of these EBOX motifs, and saw that wherever the EBOX was mutated, there was no difference among FLuc expression. But where the EBOX motif was intact, we saw a significant decrease in FLuc expression. So this suggests that the EBOXes play a role in expression for the FLuc in the HOXA9 tested transcript leader. So from this, we concluded that the EBOX is a key player in driving the promoter activity in that HOXA9 region. We also found that other HOX genes had this EBOX motif, and we tested those again using these bicistronic reporters and found that all of those HOX genes also showed false positive activity and had these EBOXes driving FLuc expression. Lastly, we moved on to analyzing data from a recent high throughput study, which tested for a potential IRES function in hyperconserved transcript leaders. So as a reminder, they found 589 hyperconserved transcript leaders and then tested over 200 of these for significant IRES activity, and found that 90 of them had a significant FLuc expression. However, now knowing that the HOXA9 transcript leader is actually misannotated, we thought this issue may have propagated through their data set. And they also never checked for promoter or splice site elements within those bicistronic reporter assays. So first, we examined a few of the genes that they labeled as hyperconserved. And to the left is the dead transcript leader, which they reported to have one of the highest IRES-like activities. And here, the transcript leader fully overlaps an ENCODE promoter highlighted in red. And RNA-seq data also aligns or does not support this long version of the transcript leader. Instead, it supports a much shorter transcript leader for the dead gene. And to the right, we have TBX1, which again, here I have a pop-in. We have overlap with the coding region, and also an example of the transcript leader overlapping a 3' splice site. So this transcript leader is also misannotated. So using promoter annotations from EPD and ENCODE databases, I next looked into that 589 hyperconserved transcript leader group to see any potential overlap for promoter elements. So wherever there was an EPD promoter, I labeled it as having some overlap. So we found that 93% of all the hyperconserved transcript leaders identified had some promoter overlap. Further, 60% of these also had enhancer overlap, so enhancer data. I next checked for 3' splice sites and found that 17% of all of the hyperconserved transcript leaders identified had an internal 3' splice site. So together, this data suggests that the IRES-like activities identified are actually explained by internal promoters and 3' splice sites rather than cap-independent mechanisms. So due to the high overlap in promoters and splice sites, I next evaluated the potential of hyperconserved transcript leader misannotation. And to do this, I used RefTSS data and their underlying K-seq data. So here, any transcription start site that fell within plus or minus 10 nucleotides of the 5' end was considered to be a 5' transcription start site. Instead, I found that only 40% of the transcript leaders identified even had a transcription start site at their 5' end. Instead, 78% of all the hyperconserved transcript leaders had a significant transcription start site peak within the transcript leader, so somewhere in the middle of the transcript leader rather than the 5' end. Next, I compared hyperconserved transcript leaders and their overlap with coding regions from alternative isoforms, and found that 43% of all of the hyperconserved transcript leaders had some overlap with coding regions. And I should also note that the Barna group excluded hyperconserved transcript leaders with high protein coding regions from their IRES or bicistronic reporter assays. So from this, the data suggests that these hyperconserved transcript leaders are misannotated and that their conservation likely is reflected by these coding region overlaps. So to evaluate the false positives in their studies, which were identified using bicistronic reporter assays, I next wanted to compare their active versus inactive groups. So for example, this was the 90 TLs they identified as having significant IRES activity versus the group they labeled as inactive. So I wanted to compare these groups and see if there was a difference in promoter elements. So here in this graph, we see that the active transcript leaders or the transcript leaders labeled as active for potential IRES activity have almost twofold more of a chance to have the promoter element. And they're 3 times as likely to have an annotated 3' splice site. This data to the right also shows that many of these active transcript leaders did not have transcription start site at their 5' end, and instead had a major transcription start site with internally. So approximately 83% of the transcript leaders had a major transcription start site within the transcript leader. I also looked at GC content compared from the active to the inactive groups and saw that the active transcript leaders had significantly higher GC content in their transcript leaders. And GC content is usually indicative of promoter elements. So together, this data suggests that the IRESes are actually false positives. So next, we tested 4 mouse transcript leaders that were labeled as active in mouse embryo tissue. So we used the bicistronic reporter assay with the SV40 promoter deleted and tested 4 of the active transcript leaders. And we found that all 4 of the transcript leaders tested showed independent F-Luke activity. And next we tested the 5' or we tested Churtle1 with 5' race to identify the 5' end. And we found from the 5' race data that it supports transcription, a transcription start site at a much shorter version of the Churtle transcript leader. So this is the annotated Churtle transcript leader, but we mapped the 5' end to this ref TSS site and EPD promoter. So this supports misannotation of the Churtle transcript leader. So if our promoter elements explained promoter IRES-like activity or explained IRES-like activities, we hypothesized that these features could be used to predict their reporter activity. So looking at different features, we included splice site data, length of the transcript leader, promoter elements, whether or not the transcript leader had an e-box, RNA-seq data, and also looked at transcription start site data. So we thought using these features that we could differentiate between those active transcript leaders or those inactive transcript leaders. And to the left is the result of the modeling I ran. So using those features, I ran a logistic regression model to classify the transcript leaders as inactive and active. And here, all of the features highlighted in green were directly correlated with a hyperconserved transcript leader being labeled active. And all of these features are correlated with a promoter element. So for example, GC content, having an internal splice site or an active e-box. So this data suggests or shows that the transcript leaders that were identified as active are most likely promoter elements. Meanwhile, features which were associated with correctly annotated transcript leaders, for example, having a transcription start site at the 5' end and having RNA-seq covering the whole transcript leader, were indirectly correlated with a hyperconserved transcript leader as being labeled as active. So here's just the results of the logistic regression model in this box form, we had an 84% accuracy from 100 trials. And this shows that the logistic regression model is using these features towards the left to predict whether a hyperconserved transcript leader would be active or not. So from this data, we find that the hyperconserved transcript leaders likely are promoter elements and do not contain IRES elements. And just as a conclusion, the HOXA9 transcript leader we identified, or which was tested for IRES activity, is actually misannotated and is much shorter than the tested transcript leader in multiple studies. The bicistronic reporter assay is also prone to false positive, so a lot of other controls must be incorporated when using these assays. We also found that USF1 and USF2 recognize a functional E-box in the HOXA9 promoter, and that most of the hyperconserved transcript leaders tested overlapped promoter elements, splice site elements, or coding regions, which may contribute to high conservation. And lastly, we found that there was no experimental evidence for the IRES expansion segment model. And this just underscores the importance of running proper controls and having a really good 5' annotations. And just as a thank you, so thank you to Joel and Gemma. Joel's my advisor, and we had a lot of interesting talks when looking at this IRES data set. And Gemma, she ran all of the experiments I talked about today. We also had some advice from the Wolford lab and Fenning lab. And again, thank you all for listening, and I can take any questions too if you have any.
Thank you so much, Christina. This was really interesting and important paper and presentation. First, I'd like to remind everyone that the recording of this will be available on the iRNA Journal Club website. So you can watch it there if you would like. Also, I have a few questions. You were looking at the 589 hyperconserved transcript leaders. Are you looking for more of those misannotated genes?
No, I only looked at the 589 group, but I'm sure that there are other misannotated transcript leaders, and it's very important, I think, that we have or we try to fix those genome annotations.
Yes, I totally agree. Reminding that if anyone has any questions, you can send them directly to the chat to me. We do have a question. If it is known what led to the misannotation of specifically HOX genes, but also hyperconserved transcript leaders, historically, like, do you know why they were misannotated?
Yeah, I think I actually, well, I can go back to this. So initially, when the HOXA9 transcript leader was annotated, they didn't really know where the AUG actually, or the gene actually was. So they just had primers in this region and used like primer walking backwards using RT just to go back. And I think they actually got into this fusion transcript region. So they really identifying this intron. So I think that's how they first got this longer version. So this was in 1998, and it was just never, I guess, originally when they were looking at these papers later on, they didn't check whether it aligned with other data too.
I see. So, all right. So I guess ENCODE just released their version 41, is it? Do you know if it's corrected there? You haven't checked?
Yeah, I'm not sure. I haven't checked the newest version. But I think if you look at the, it's just important to check whether you are actually seeing, for example, the RNA-seq data and transcription start sites over the transcript leaders you're using.